Today, I'm going to talk a little bit about my work at looking at RNA modifications in the human transcriptome. So first off, what is an RNA modification? It is a chemical alteration to a canonical nucleobase. These can be simple additions of a methyl group or complicated multi-ring structures added to the base or the sugar backbone. And they can be internal to RNA strands or on the ends. And so here's just a few examples of some fairly common ones found in human mRNA, like M6A and inosine and pseudouridine. These RNA modifications are involved in almost every aspect of the transcript regulation, and dysregulation of the modification patterns can lead to diseases and have been implicated in diseases such as cancer. They're involved in microRNA processing, translation, transcription, degradation, basically all aspects of these RNA life cycles. And there's a lot of them. To date, we've discovered about 170 naturally occurring RNA modifications through the three major branches of life. And then there is some overlap between these three branches. And they can be very complicated or very simple modifications. These have primarily been detected using mass spectrometry methods or short read sequencing coupled to an assay specific for an individual modification. So using like an antibody to pull down the modification, or a specific chemistry that interacts with the modification specifically, or behavior of the reverse transcriptase with that modification in your sequencing data. But this is one assay, one modification. And so with nanopore sequencing, with direct RNA, the modifications are kept intact because we don't go to cDNA. And so these modifications, as they pass through the nanopore sensor, they cause ionic current changes into the signal that are different from the canonical nucleotide. And so we can detect these differences in the ionic current using software packages like Nanopore, which was written as part of my group from a previous postdoc, Adrian Leger, who is sitting in the audience now. And so this software compares these two samples using a Gaussian mixture model, and then clusters the data to then find the spots where there are positions where there's significant differences. But this is a comparative method. So you have to have a sample with the modification and a sample without the modification. Traditionally, most people use a wild type sample and then they manipulate the writer enzyme for a particular modification, either using a knockout or a knockdown experiment or a chemical inhibitor. And so this causes you to actually derive your RNA from two separate cell lines, the modified and the unmodified form. And you're limited to the modifications that are dependent on that disruption. Instead, what I'm going to be focusing on today is a different strategy where you have one cell line and you generate an in vitro transcribed copy of the transcriptome where you erase the signal of auto modifications. And so you can detect basically all modifications at the same time with a single sample derived from the same cell pellet. And so briefly, this is a diagram of how this process works. First, we isolate our total RNA, and then we enrich for poly-A RNA. Some of that is then directly sequenced using the native RNA sequencing. And then for the other half, we go through a cDNA synthesis step using a template switching oligo to add the T7 promoter to the 5' end of all of our transcripts. And then we synthesize the RNA in vitro from that T7 promoter using the T7 polymerase. And this will make a collection of now modification-free RNA transcripts. And then because the T7 polymerase has a tendency to make some incomplete transcripts and doesn't make everything full length, we in vitro polyadenylate so that we can sequence with Nanopore. And then we use Nanopore to compare the IVT sample to the wild type sample. And so with this experiment, we isolated RNA from K562 cells. And with our native libraries, we got about 9 million reads from each of these and an RNA-002 promethine flow cells. And then with our IVT, we got 5.4 and 2.9 million reads. This allowed us to analyze about 2.9 million positions within the human transcriptome. We found 26,000 RNA modifications from 2,500 transcripts and 1,700 genes. And so we do have some alternative isoforms that we can detect here. And when we look at the distribution of the significant sites we find with Nanocompore across the metagene, and so this is looking at averaging across every position within all transcripts. In red, we have the sites that we found from Nanocompore, and then in blue and green are annotations of where either all annotated modifications or just M6A. And for the most part, our sites overlap pretty well with the annotations, but we are finding more in the coding region comparison to the annotation, which kind of makes sense because we're finding things that are less well annotated. And so here's just looking at some of the motifs in k-mers that we found from this analysis. And so this is a Silomar analysis where we ranked the k-mers that we found that were significant from the most significant to the least significant p-value from Nanocompore. And then we have the hypergeometric p-value here on the y-axis. And I'm just highlighting here the top 10 motifs that we found. And 3 of them are a DRAC motif, which is the classic M6A writer. And 4 of them are motifs that are associated with M5C. And then there's one that is like an off by 1 DRAC motif, but we can't quite call that a DRAC. And so we are getting an enrichment for M6A and M5C signals. And here's just one example of a classic cancer gene in the BCR. So this K562 is a leukemia cell line. And so this is classic for driving those leukemia cancers. And I have on top the gene transcript model from the 5' end to the 3' end. And this is just highlighting a 181 base pair region at the 5' end in the first exon of this transcript. And I'm annotating from RM base these proteins that are associated with RNA modifications. And for the most part, we're getting good alignment with the M6A signal, which I've highlighted in red, the DRAC motifs. And then in black is our Nanocompore significant sites, and so we have 6 here. But right next to 2 of these M6A sites, we're finding a motif associated with M5C, which is lining up right with our M6A, or our Nanocompore sites. And so we think that there might be some coupling of M6A and M5C here. And when we do a chi-square looking at coordination for different sites across the transcripts, most of them are not significant, which actually matches the current expected model that these modifications are deposited randomly. However, we found 3 that were significant for this coordination. And so I'm going to show you an example of just this one in the middle here. And so this is KRT-CAP2. And I have the transcript model now from the 3' end to the 5' end. It's the reverse orientation gene. And we have 3 significant sites found from Nanocompore. One here at the 3' end, which looks to be a metal 3 dependent M6A site. And then the 5' end, it looks like we have a metal 16 dependent M6A coupled fairly closely to a M5C site. And when we start looking at the phasing of these modifications, because we have single molecule information, we can look on the same molecule, are they modified together or on different strands? And so we have positions 403, 165, and 154, which would be 1, 2, and 3. And then if you look at, I have the modification pattern encoded. 0 would be unmodified, 1 is modified. I then have the observed counts from the sequencing, and then the expected counts that is calculated from the linear probabilities of the combination of the modifications. And what stands out from here is that the 2 modifications that are next to each other near the 5' end and exon 2 are above expectation when they're co-modified and below expectation when they're opposite. So if you look at the 3rd row down where it's 0, 1, 1, we have 111 counts in the observed compared to 35. Where if you look at the 2 down below that, we have 0, 1, 0, we have 94 in the observed and 165 in expected. So when they're opposite, they're below expectation. And so with that, I just want to conclude that the IVT reference is useful for detecting multiple different types of RNA modifications at the same time. But analysis of this type of information is a lot more challenging without using alternative information to infer the identity of the modifications. And then as observed by others in the field really recently, that there does seem to be some coordination between M6A and M5C. And we're now following up these observations with orthogonal validation techniques. And science is not done in isolation. I have been working with a wonderful group of people in Italy and in Cambridge. And I really want to thank the wonderful group that I've been working with to make this talk and project possible. And a big thank you to Lucia, who is the experimentalist who generated all of these data.